Hello everyone, my name is Ankur and welcome to RJPT Simlab simulation. In this video, we will learn about to conduct a study on muscle relaxant properties of diazepam, specifically its effects on muscle grip strength using a Rota-Rod's apparatus in mice. At the end of this video, we will be able to tell that how does a Rota-Rod apparatus is used to study about the therapeutic effects of diazepam by using mice. Now let us start with the video. At first, let us see the introduction part. The introduction says diazepam, a member of benzodiazepine class of drug, is widely used for its anti-anxiety and muscle relaxant properties. That, as we know, the diazepam is a member of benzodiazepine class. So the benzodiazepine is the class of drug of diazepam, and it is an anti-anxiety drug and is also used for its muscle relaxant properties. The study of these properties, particularly the muscle relaxing effect, is crucial in understanding the drug's therapeutic potential. So, this muscle relaxing effect of benzodiazepine is very important for to understand the drug's therapeutic potential. This experiment aims to investigate the muscle relaxant properties of diazepam, specifically its effect on muscle grip strength, using a Rota-Rod's apparatus in mice. So, in this experiment and study, we will learn about the muscle relaxant properties of diazepam, and by studying its muscle grip strength by using mice through a Rota-Rod's apparatus. Now let us see the equipments required for this experiment. Our first apparatus is Rota-Rod's apparatus, then weight machine, syringe and mice cage. Then the subject we require is mice, which should be of 20 to 30 grams. Then the drugs which we require are is diazepam, and we are using 0.4 milligram per milliliter. Let me tell you more about Rota-Rod's apparatus. As you can see, this is the image of Rota-Rod's apparatus. As we can see here, these are the sections over which we will place our rat or mice. As we can see, this is our mice. Two mice are placed between the sections, and this is the Rota-Rod's. This is the apparatus rod over which we will place our mice. As we can see, this is the rat placed over the rod. And then when the rotation starts, as we can see, this is the conveyor belt which is used for the movement of this rod. It circulates and moves, and due to its circular movement, the rats try to stay on the rod, and that's how we learn about the grip strength of the rat. So the principle of the Rota-Rod's apparatus says that the principle of Rota-Rod apparatus is to measure an animal's motor coordination, balance, and endurance by timing how long it can stay on the rotating rod before falling. The principle says that it measures its motor coordination. As we know, motor coordination are the nerves signaling on the nerve cells, the balance and the endurance by timing. How long does a rat stays on the rod? How long it can stay on the rotating rod before falling? As we can see, these are the rats that have fallen. Then, how does we analyze the data? This fall time, as we can say, the time at which the rat falls, is a direct measure of motor function. A shorter fall will indicate poor more motor coordination, while a longer fall time indicates better performance. So, as we can see, if the rat falls in a very short time, that indicates poor motor coordination, but if the rat falls in a longer time, that indicates better performance. Now, let us continue our experiment. Now, let us see the principle of this experiment. The principle of this experiment lies in the pharmacological action of diazepam. The drug induces muscle relaxation along with a calming effect, which helps reduce anxiety and tension. The loss of muscle grip, as observed when a mouse falls from the rotating rod of the Rota-Rod apparatus, is an indication of the muscle relaxation. The time taken by the animal to fall from the rotating rod is taken as an index of muscle relaxation. So the principal part says that the experiment's principle lies on the pharmacological action or therapeutic action of diazepam. The drug induces the muscle relaxation. So we are studying about the muscle relaxant property of diazepam. So this drug relaxes the muscles along with a calming effect, which helps in reduces the anxiety and tension of the animal. So due to that, the loss of grip happens of the animal is observed, and when the mouse falls from the rotating rod apparatus, that is an indication of the muscle relaxation. So when the muscle starts to relax, the rat loses its grip and falls from the rotating rod. And the time taken for the animal to fall from the rotating rod is the index of the muscle reaction, or is the main part of the muscle relaxation. Now this is the observation table of this experiment. As we can see, which shows the body weight, treatment, the drug we are using, then the before drug observation fall time and after drug fall time, and then percentage and decreasation of the time. We are using six mice for this experiment so that we can observe very easily the differences of the time. Now let us see the procedure part of this experiment. The first, we have to organize the mice into a group of six. Then we have to assign a number to each mouse after weighing them. Then we have to activate the Rota-Rod apparatus and set the speed to an optimal range of 20 to 25 RPM. So we have to set the speed at 20 to 25 rotations per minute. Then place each mouse on the rotating rod one after the other. The apparatus has six compartments, so each mouse should be in its own compartment. As we have seen in the earlier in the image of the apparatus that it has compartments, so we are using the Rota-Rod apparatus of six compartments. That's why we are using six mices. Then we have to place one by one each, and then we have to start one by one. Then we have to record the fall time, the time when the mouse falls from the rotating rod. And after this, switch off the Rota-Rod apparatus and return all the mice to the original cage. Then we have to select each mouse and administer through the syringe via the syringe, and then move it to the empty cage. Dispose of the used syringe in the trash bin after application. Then, with the help of a syringe, we have to administer the diazepam drug. Then we have to allow a waiting period of 30 minutes. We have to wait for 30 minutes so that the drug could take its action. And after that, we have to reactivate the Rota-Rod apparatus and maintain the speed within the 20 to 25 RPM range. Then after that, once again, place each mouse on the rotating rod in a sequential manner, similar to before, and ensure each one is on its own compartment. Then we have to record the fall time again and switch off the Rota-Rod apparatus and return all the mice to the original cage. And then we have to analyze them and compare the fall of time of the mice before and after the diazepam's treatment. And that's how we will differentiate between the mice after the drug administration fall time and the fall time before the drug administration. And that's how we will study about the therapeutic effect of diazepam. Now, let us perform this experiment. And for that, we are using our RJPT's Informatics Simulatory Laboratory. As we can see, this is our laboratory where we will perform our experiment. This is our table, and this is the Rota-Rod's apparatus. As we can see here, this is the rotating rod which is connected to the motor inside this chamber, and they are connected via a cable. As we can see, this is the conveyor belt or cable. This is the cable which rotates this rotating rod. And we can see the six compartments inside that, which are separated by this glass wall. 1, 2, 3, 4, 5, 6. Six chambers for six different mices. And this is the timer. This timer runs where we have to start each timer after placing the mice in them. Now, this is the UI interface of our simulatory laboratory. As we can see on the left side of the toolbar, we can see the cage and the weighing machine button. On the right side, we can see the syringe. By clicking them, we can place them over the laboratory's table. On the bottom side, we can see the instruction part, which shows the procedure part according to which we will perform our experiment. On the right side of that, we can see the next and skip button so that we can directly move and jump on to the next step. On the top side, we can see the observation table drop down. As we can see, this is the observation table that we have seen earlier. Six mice, so six serial numbers, body weight. We have to weigh each mice and then we have to write the body weights of each mice. Then the treatment, we are using the diazepam drug, and the fall off time before drug and after drug in seconds that we will calculate from the timer, and the percent in the decrease in the time. By clicking this procedural flow button, we can see the flow chart of the procedure. We can see here by scrolling this, and by clicking each and every step, we can directly jump on to that step. So now let us perform this experiment. For that, we will follow our instruction which says place the cage from the left sidebar. So we'll click this cage, and as we can see, our the cage filled with six mice are placed on the table. And now we have to place the weighing machine from the left sidebar. As we can see, we have placed it over here. And now mark and weigh mice one. We will start weighing. We will open the cage and we will take out our very first mice and mark it with the help of a marker. The single one line shows indicates first number of mice. We have weighed it, and now we have placed it back to the cage. Now we will weigh our second mice. We will take it out and we will draw two lines which indicates second mice. We will weigh it and we will place it back inside the wing, inside the cage. We will take out the third mice and we will draw three lines with the help of the marker. As we can see, one, two, and three. Three line indicates third number mice. And again, we will place it inside the cage. Similarly, for the fourth mice, we will wait. We will slowly take it out so that the mice does not get scared. We will draw four lines to show fourth number, and we will weigh it from the weighing machine. And after that, again, we will place it inside the cage. Now, similarly for the fifth mice, we have to weigh all the mice very carefully. As we can see, the body weight of each mice has been written on the observation table automatically. The first mice weighed 20 grams, second 25, third 26, fourth 21, and the fifth we have weighed it currently which has 23 grams weight. And now we will weigh our last mice. We will carefully take it out from the cage and we will draw six lines which will indicate sixth number mice. And now we will weigh it from the weighing machine which says 24 grams weight of the mice. And again we will place it back inside the cage very carefully and slowly. And now moving forward, we will now take each mice and put in the Rota-Rod for the reading. So now we will click the take mice button and now we will start the machine. So through that, the Rota-Rod has started to move. As we can see, we have placed the mice one by one. Sequentially, we will place the mice on the rotating rod and we will wait for it to fall. And and by placing each mice, we will start its timer. Six compartments has its each separated timer. As we can see, and we have to start each timer after placing the mice. As we can see, the timer has started for the first mice, for the second, third, fourth, fifth, and sixth. The rats are moving over the rotating rod, as we can see here, and they are struggling to move. We will wait for them to fall from the rotating rod, and then we will stop the timer for each mice. As we can see here, they are taking a longer period of time to fall from the rotating rod, which means that is the normal timer for the mice to fall. When the muscle relaxation will takes place, the mice will lose its grip like this, as we have seen here. The second, third, the first also. The timer stops automatically. The fourth one has also fallen. The fifth one, and similarly, as we can see here, the sixth one has also fallen. Now, after this, we will place all the mice again back inside the cage. And now we will place the syringes from the right side bar. We will click the syringe, and as we can see, six different syringes for six different mice. We will apply diazepam to each mice. We will carefully take out the mice, and through the intraperitoneal route, we will apply the diazepam to the mice. As we can see here, carefully we have to administer and apply the syringe to the mice so that it does not harms the mice. One by one, slowly, we will apply the syringe very carefully. And again, we will place it inside the cage. On the observation table, we can see the fall of time before the drug. As we can see here, the first mice took 66 seconds, second took 60, third 58, fourth 64, fifth 62, sixth 61 seconds. So the average fall of time before giving the drug is 61.83 seconds. Now we will apply our diazepam to our last mice. We will apply it very carefully and very carefully we will place it inside the cage. And after placing it inside the cage, we will wait for 30 minutes to take the readings. As we can see, the countdown has started. We can we can skip it by clicking the skip button. And now we will take each mice and put it inside the Rota-Rod for the reading. Now after the diazepam, and we have waited for 30 minutes to let diazepam take its effect. And now we will place each mice inside the Rota-Rod similarly. We will switch on the Rota-Rod apparatus. We have set the RPM, which is 22. It should be between 20 to 25 RPM. So now we will place our first mice and we will start our reading, our timer. And as we can see here, the timer has started. And after starting the timer, in a few seconds, the rat falls. It loses its grip and it falls. Similarly, for the second mice also, for the third mice too, as we can see here. For the fourth mice, we have placed our fifth mice, and now we have placed our sixth mice. Our fifth mice and fourth mice both have fallen, and our last sixth mice has also fallen, and we have stopped the timer. Now we'll place all the mice inside the cage again. And now after the drug administration, we have recorded all the readings of the six mice. The first mice took 5 seconds, second 8, third 10, fourth 3, fifth 5, and sixth 8 seconds. So the average fall of time after the drug administration is 6.5 seconds. So as we can see by just reading the observation table that there is a decrease in the time after administration of the diazepam drug, which says that and shows that the diazepam has shown its therapeutic effect of the muscle relaxation, and by calming the animal loses its grip and falls from the Rota-Rod's apparatus, which shows that the muscle relaxation has took very quickly. This column shows the percent decrease in the time. So as we can see here, this is a decrease in the time. By clicking the Save Observation Table button, we can save our observation and the readings. As we can see here, this is the data that we have collected and written on the observation table. And by writing our name over here and our roll number, we can print it. As we can see, we can print it and we can save it in our system. Now coming back to the experiment, here we have completed our experiment. And now this is the result part. As we can see, the percent decrease for the first rat from 66 to 5 seconds. In percentage, we can say that it is 92.42 seconds. For the second mice from 60 to 8 seconds, the decrease in time in percentage is 86.60%. For the third mice from 58 to 10 seconds, it's 82.75%. For the fourth from 64 to 3 seconds, the percentage decrease is 95.31 percentage. For the fifth mice from 62 seconds to 5 seconds, the decrease in percent is 91.93%. And for the sixth mice from 61 seconds to 8 seconds, the decrease in time in percentage is 86.88%, which shows the decrease in the fall of time of the animal, which shows that the diazepam has shown its effect. The result says the outcome of the experiment will show how diazepam affects the muscle relaxation. So we have observed that, and if the mice take shorter time to fall off the rotating rod after being given diazepam, as we have observed, it shows that the drug has muscle relaxant effect. So we have observed that the diazepam drug, our diazepam drug, has muscle relaxant effect. However, the results can vary based on the specific conditions of the experiment and individual responses of the mice. The result can also vary according to the condition of the mice, but our all the mice were very healthy and we have done in a very safe condition. So our result is also very valid. So here we have studied about the effect of the diazepam, its muscle relaxant effect. Here we have completed our experiment and we have come to the end of this video. I hope that you have learned a lot in this video about the effect of diazepam and by how using the Rota-Rod's apparatus for the mice. So now, thank you guys for watching this video. I hope that you have learned a lot in in this video. And for more such learning content, subscribe to our YouTube channel RJPT Informatics. You can also follow us on our LinkedIn profile, on Instagram, as well as in Facebook. We update and upload our such learning content over these platforms. Thank you.